You think pressure is about weight. You think it is about force pushing down. 7,000 meters below the surface in the Mariana Trench, the pressure is not 1,100 times what you feel right now, standing wherever you are. It is 1,100 atmospheres, crushing every square inch of anything that dares exist there. That is 16,000 pounds per square inch. That is the weight of 50 jumbo jets balanced on your thumbnail. Your skull would crack, your lungs would collapse into wet tissue, your bones would splinter like dry wood. And yet, down there, a pink, translucent fish swims. It hunts, it breeds, it thrives. The Hadal snailfish, Pseudoleparus swearii, exists where you cannot, and the reason it survives is stranger than any science fiction you have read. It is a conspiracy of biology, chemistry, and physics working together in ways scientists only started understanding in the past 3 years. What makes these creatures survive is not simply thick skin or strong bones. It is molecules that bend the rules of physics. It is membranes that should fall apart but do not. It is genes that stopped working millions of years ago on purpose. And it is a compound that smells like rotting fish but keeps proteins from collapsing under pressure that would liquefy steel. Also, this script is researched and factual, using sources from Nature, Ecology, and Evolution published in April 2019, Science published in June 2024 on homeocurvature adaptation by Winikoff and colleagues, and Communications Chemistry published in September 2022 by Dugan and team at the University of Leeds on TMAO water structure resistance under 4 kilobar pressure. These are peer-reviewed studies with direct experimental evidence, not speculation. Now back to the information. That is 36,000 feet, over 1,000 atmospheres. At that depth, water itself changes. The molecules pack tighter. The hydrogen bonds between them distort. This is not just squeezing. This is fundamental alteration of how matter behaves. A study published in Communications Chemistry in September 2022 showed that at 4,000 bar pressure, water molecules lose their normal tetrahedral network structure. The second solvation shell, the layer of water molecules around other water molecules, starts to collapse. For anything alive, this is catastrophic. Proteins need water to maintain their shape. Enzymes need water to catalyze reactions. Cell membranes need water to stay fluid. When pressure changes water structure, life stops working. In 2019, Chinese researchers led by Kun Wang from Northwestern Polytechnical University published the first complete genome of Pseudoleparus swearii in Nature, Ecology, and Evolution. They collected 4 specimens from 7,000 meters down in the Mariana Trench using remotely operated landers. The fish took 3 hours and 11 minutes to reach the surface. When the team examined them, they found something immediately strange. The skull was not closed. There were gaps, literal holes where bone should have been. Wang explained that these gaps allow internal and external pressures to balance. A complete skull would be crushed. An incomplete skull flexes. In the Hadal snailfish, the gene has a frameshift mutation. It terminates early. Bones do not fully calcify. They stay flexible. Cartilage can compress and bend under pressure. Hard bone cannot. At 7,000 meters, rigid equals dead. Flexible equals alive. This is not a minor adaptation. This is the skeleton itself being rewritten. The reason is not aesthetic. Pigments are expensive, making them costs energy. In the Hadal zone, food is scarce. A whale carcass falls from above maybe once a year. A dead fish drifts down occasionally. You cannot waste energy on color when there is no light to see it and no calories to spare. The transparency also serves another function. The skin is permeable to water. This helps equalize pressure across cell membranes. If skin were impermeable, pressure differentials would develop. Cells would rupture. By allowing water to pass through, the Hadal snailfish keeps internal and external environments similar. When bacteria break down TMAO, they produce trimethylamine, TMA, that is the rotting fish odor. But in living deep sea fish, TMAO does not rot. It protects. Fish at 3,000 meters have 120 millimoles. The Hadal snailfish at 7,000 meters has 261 millimoles per kilogram. That is over 6 times the concentration of shallow fish. This is not random. This is a direct response to pressure. The molecule was called a piezolite, a pressure salute, but no one understood the mechanism. How does a small organic molecule with 3 methyl groups attached to a nitrogen oxide actually stop proteins from unfolding under 1,100 atmospheres of force? A study published in September 2017 by Cristina Papini and colleagues in the journal Biophysical Journal tried to answer this. They measured the effect of TMAO on protein volume changes under pressure. The results surprised them. TMAO did not directly affect the volumetric properties of proteins. It did not change how much proteins compress under pressure. Instead, it increased protein stability indirectly. It made proteins harder to unfold at any pressure by increasing their overall thermodynamic stability. It was not specifically evolved to counteract pressure. It was just a strong stabilizer that happened to work under pressure. But that conclusion was challenged 5 years later. In September 2022, Lorna Dugan at the University of Leeds published new findings in communications chemistry. Her team used neutron scattering and computational modeling to analyze water structure at 4 kilobar pressure, with and without TMAO. What they found changed everything. TMAO does not just stabilize proteins, it stabilizes water itself. TMAO forms strong hydrogen bonds with water through its oxygen atom, and this preserves the network even under crushing pressure. Dugan's team calculated an osmolyte protection ratio, the ratio at which pressure-induced energy changes and TMAO-induced energy changes cancel out. At 100 millimoles of TMAO per kilogram, the protection ratio matches a depth of around 1,000 meters. At 261 millimoles, it matches 7,000 meters. The concentration of TMAO in deep-sea fish is not random. It is calibrated. Evolution has tuned the exact amount needed to keep water functional at specific depths. For years, scientists assumed TMAO would keep rising with depth. It does not. Fish living below 6,000 meters in the Hadal zone have TMAO concentrations that plateau. They do not exceed 300 millimoles per kilogram. This suggests a limit. Either producing more TMAO becomes toxic, or the fish are using alternative mechanisms. Researchers from China's deep-sea exploration programs found a mutation in the RTF, one gene in all deep-sea fish below 3,000 meters. This gene boosts transcription efficiency. It helps cells function under crushing pressure. The mutation is absent in shallow fish. It is present in every Hadal fish examined. This is a second mechanism, independent of TMAO. This one was discovered only in June 2024. Jacob Winikoff, a researcher at Harvard University and the Monterey Bay Aquarium Research Institute, published a study in Science on comb jellies, ctenophores. These are not fish. They are gelatinous predators that live at various depths. Winikoff collected 17 species from depths ranging from the surface to 4,000 meters. He analyzed their cell membranes. What he found was shocking. In shallow water animals, plasmalogens make up maybe 10% of membrane lipids. In deep sea ctenophores, they make up 75%. 3/4 of the membrane is curved lipids. Channels close, transporters freeze, the cell dies. But curved lipids behave differently. Under pressure, the curvature flattens out. The molecules become straight. The membrane stays fluid. Winikoff called this homeocurvature adaptation. The lipids are curved at surface pressure so that they become the right shape at deep sea pressure. It is like pre-bending a metal bar so that when weight is applied, it straightens to the perfect angle. Winikoff's team tested this. They took Escherichia coli bacteria, common gut bacteria that normally live at surface pressure. They genetically engineered some of the bacteria to produce plasmalogens by inserting a gene cassette from Clostridium perfringens. Then they put both normal and plasmalogen-producing bacteria in high-pressure chambers, 480 bar pressure. The normal bacteria died. The plasmalogen bacteria survived. Producing curved lipids made them pressure tolerant. This was direct experimental proof that lipid shape matters for pressure survival. When deep-sea actinophore tissue was brought to surface pressure, the membrane structure inverted. The lipids flipped from a bilayer, which is 2 sheets of lipids forming a membrane, to an H2 phase, which is tubular lipid structures. The membrane literally turned inside out. The cells could not hold together. The animal dissolved. For over a century, marine biologists have known that deep-sea creatures brought to the surface often disintegrate. Even when handled carefully, even when kept cold, they fall apart. The assumption was always mechanical damage, but it is not mechanical, it is chemical. The membranes are held together by pressure. Without pressure, they invert. The lipid curvature that is correct at depth becomes wrong at the surface. The same adaptation that keeps them alive below kills them above. They are interlocking systems. Remove one, and the others fail. The Hadal snailfish does not survive because of one adaptation. It survives because of 5 working together. Why does fish life stop around 8,000 meters? A study published in 2014 by Paul Yancey in PNAS proposed an answer. It is TMAO. Beyond a certain concentration, TMAO becomes toxic. At around 300 millimoles per kilogram, the internal osmolality of the fish, the concentration of dissolved solutes, reaches parity with seawater. Seawater has an osmolality of 1,100 millismoles per kilogram. Fish normally maintain internal osmolality around 350 millismoles. The difference drives osmoregulation. Water flows in, ions flow out, the fish stays balanced. But if TMAO keeps increasing, the internal osmolality rises. At around 8,000 to 8,500 meters, the fish's internal osmolality would equal seawater's. The osmotic gradient reverses, water flows out, the fish dehydrates. Even if it could tolerate the pressure, it would die of osmotic stress. Extrapolating the data, they calculated that at 8,200 meters, the osmolality would reach isosmotic state, equal to seawater. Beyond that depth, fish physiology cannot function. This is not a hard wall, it is a biochemical constraint. Fish are limited not by pressure crushing their bodies, but by the protective molecule they need to survive becoming the thing that kills them. The skeleton is not just about flexibility, it is about density. Most shallow fish have heavy, calcified bones. They also have swim bladders, gas-filled sacs that provide buoyancy. Inflate the bladder, and the fish rises. Deflate it, and the fish sinks. This works in shallow water, it fails in deep water. At 3,000 meters, the pressure is 300 atmospheres. Filling a swim bladder with gas at that pressure requires enormous energy. Most deep-sea fish do not have swim bladders. Instead, they use other methods to stay neutrally buoyant. Some have fatty livers. Fat is less dense than water. A large liver filled with lipids provides lift. Others, like the Hadal snailfish, have gelatinous tissues. The body is soft, watery, and low density. Combined with thin, cartilaginous bones, the fish is barely denser than seawater. It floats without needing gas. In 2019, the Nature Ecology and Evolution study on Pseudoleparus swirei found that the fish's bones are not just cartilaginous, they are undermineralized. The gene responsible for bone mineralization, BIGLAP, has a frameshift mutation. Another gene, TEMM251, which also plays a role in bone formation, is completely lost. A third gene, TEMM263, is gone. These are not accidental losses. They are evolutionary deletions. The fish's genome actively prevents bones from hardening. This is the opposite of what you expect. On land, strong bones are essential. In the deep sea, strong bones are a liability. They weigh you down, they crack under pressure. Weak bones, flexible bones are better. Its jaw muscles are weak because the skull cannot support strong muscles. So it hunts differently. It scavenges. It waits for food to fall from above. It eats amphipods, tiny crustaceans that live on the seafloor. It swallows them whole. The stomach is inflated, oversized. It can hold large amounts of food when food is available. This is necessary because food in the Hadal zone is rare. A single meal might need to last weeks. The eyes are another adaptation. Most deep-sea fish have large eyes to capture what little light exists at mid-depths, but the Hadal snailfish lives below 6,000 meters. No sunlight reaches that far. Bioluminescence is the only light, and it is rare, so the Hadal snailfish has reduced eyes. They are small, non-functional. The fish does not respond to light. In the 2019 study, researchers shone bright lights at captured Hadal snailfish. The fish did not react. Genomic analysis revealed why. Several photoreceptor genes are lost. The LWS gene, which detects long wavelengths like red light, is gone. The RH2 gene, which detects central wavelengths like green light, is gone. The SWS2 gene, which detects short wavelengths like blue light, is present but barely expressed. Only the RH1 gene, which is critical for monochromatic vision in very dim light, is active. This suggests the fish might have residual ability to see faint bioluminescent flashes, but nothing more. The Hadal snailfish has 3. This means it produces more otoliths more efficiently. At 7,000 meters, forming calcium carbonate structures is difficult. Pressure inhibits crystallization. Having multiple gene copies compensates. The fish maintains functional hearing even where chemistry says it should not be possible. This gene encodes ferritin, a protein that stores iron and protects cells from oxidative stress. High pressure increases production of reactive oxygen species, unstable molecules that damage DNA and proteins. Ferritin neutralizes these. By having 14 copies of the gene instead of the normal 2 or 3, the Hadal snailfish produces massive amounts of ferritin. It is constantly defending against oxidative damage. This is not a passive adaptation. This is active biochemical warfare against pressure-induced cellular harm. If the fish burned fat too quickly, it would starve. By slowing lipid metabolism, the fish extends how long it can survive between meals. Another gene, CPT1, which is involved in fatty acid transport into mitochondria for energy production, is upregulated. This seems contradictory, but it is not. CPT1 increases when fatty acid oxidation is needed. The fish is not burning fat constantly. It is burning it efficiently when it needs to. In September 2024, a study published in Water Biology and Security by Han Xu and colleagues from the Chinese Academy of Sciences examined the liver of Pseudoleparus swirei in detail. They used lipidomics and proteomics, techniques that analyze all lipids and proteins in a tissue. The results were striking. The liver is enlarged. It contains high levels of cholesterol esters and triacylglycerols, storage fats. These provide energy during prolonged fasting. The liver also has elevated coenzyme Q and ATPase, molecules that enhance energy production efficiency. When food is available, the fish eats. It stores fat in the liver. When food is not available, it burns the fat slowly, efficiently, using coenzyme Q and ATPase to extract maximum energy. The proportion of unsaturated fatty acids is significantly increased. Unsaturated fats have double bonds in their carbon chains. These bonds create kinks. The kinks prevent lipids from packing tightly. This keeps membranes fluid at high pressure. Cholesterol levels, on the other hand, are reduced. Cholesterol normally stiffens membranes. In the deep sea, membranes need to be more fluid, not less, so cholesterol is minimized. Phosphatidylethanolamine, another lipid that increases membrane rigidity, is also reduced. The liver's membrane composition is fine-tuned to maintain fluidity under 1100 atmospheres of pressure. The liver is not just a storage organ, it is a defense system. It protects the rest of the body from oxidative stress. Without this defense, pressure would slowly kill the fish through cumulative cellular damage. They compared it to Tanaka's snailfish, Liparis tanaki, a shallow water relative that lives in tide pools. The two species diverged 18 million years ago. Tanaka's snailfish stayed shallow. The Hadal snailfish moved deep. Over 18 million years, the deep-sea fish accumulated specific genetic changes. 33 genes are unique to Hadal snailfish. 19 genes are pseudogenes, non-functional remnants. 51 genes present in other snailfish are absent in Hadal species. This is not just adaptation. This is genomic restructuring. The trenches are geographically isolated, but the fish managed to colonize them independently. This means the adaptations for deep-sea life evolved at least twice, possibly more. Convergent evolution, different populations, same solutions. Some species are depth-constrained. They can only live deep. If brought to the surface, they disintegrate. Other species are depth-flexible. They can live at multiple depths. The difference is lipid curvature. Depth-constrained species have extreme negative curvature lipids, 75% plasmalogens. These membranes are only stable under high pressure. Depth-flexible species have moderate curvature lipids, 40 to 50% plasmalogens. Their membranes work at both high and low pressure. This suggests that evolution is trading off specialization versus flexibility. Extreme adaptation to deep pressure makes you vulnerable at the surface. Moderate adaptation lets you move between depths, but you're not optimized for either. They modeled lipid bilayers under pressure using molecular dynamics. They calculated the energy required to maintain bilayer structure at different pressures. For shallow water lipids, increasing pressure increases the energy cost. The bilayer becomes unstable. For deep sea lipids, the opposite happens. Increasing pressure decreases the energy cost. The bilayer stabilizes. At surface pressure, deep sea lipids are unstable. At 4,000 meters, they are stable. The simulations match the experimental data. The homeocurvature model works. Lipids are pre-curved so that pressure straightens them to optimal shape. Fermentation under pressure. Bioreactors for deep sea mining. Microbial fuel cells in the ocean. The principles learned from comb jellies and Hadal snailfish could let us create pressure-tolerant organisms for human use. It is not science fiction. It is applied biochemistry. The TMAO limit is one answer, but is it the only answer? A 2022 study published in the Journal of Chemical Physics by Angelina Folberth and Nico van der Vecht explored this. They simulated TMAO under extreme pressure using molecular dynamics. At 10 kilobar pressure, 10,000 atmospheres, TMAO changes. Its dipole moment increases. It binds more water molecules. It shifts from 3 water molecules coordinated around it to a partial fourfold coordination. This changes how TMAO interacts with proteins. Specifically, it strengthens hydrophobic interactions. Hydrophobic parts of proteins, which normally repel water, become more tightly folded in the presence of high-pressure TMAO. This stabilizes proteins further. It makes them too rigid. Enzymes need flexibility to function. If TMAO locks them in place, they stop working. This is why TMAO is never found in high concentrations without a destabilizer. In sharks and rays, which have high TMAO levels, they also have high urea levels. Urea destabilizes proteins. TMAO stabilizes them. Together, they balance. But deep-sea fish do not use urea. They use TMAO alone. So there is a concentration limit. Beyond 300 millimoles per kilogram, TMAO likely becomes toxic. It does not swim fast. It does not chase prey. It waits. It conserves energy. Over millions of years, this slow life has resulted in fewer genetic changes. But fewer genetic changes mean slower evolution. If environmental conditions change, the Hadal snailfish cannot adapt quickly. It is locked into its current niche. They found up to 23 mature eggs per female. Each egg was unusually large, up to 9.4 millimeters in diameter. Large eggs contain more yolk. More yolk means the larvae can survive longer without feeding. But large eggs require more energy to produce, and there are fewer of them. A shallow water fish might produce thousands of eggs. The Hadal snailfish produces 2 dozen. This is a trade-off: quality over quantity. Each egg has a better chance of survival. But if something goes wrong, if the eggs are eaten or damaged, the fish cannot replace them quickly. Reproduction in the deep sea is slow, expensive, and risky. If the adults live at 7,000 meters, why do the larvae go up? One hypothesis is that shallow water has more food. Larvae need to grow quickly. The deep sea does not have enough food to support rapid growth, so the larvae migrate upward, feed in the mesopelagic zone, then return to the deep as adults. But this requires surviving pressure changes. The larvae must tolerate going from 7,000 meters to 1,000 meters and back. That is a 6,000 meter, 600 atmosphere pressure change. How do they do it? As they mature, they produce more TMAO, their bones remain cartilaginous, their membranes shift to higher plasmalogen content, and they move deeper. This is speculative. No one has tracked the full life cycle of a Hadal snailfish, but the evidence suggests vertical migration during development. Incomplete cartilaginous skulls that equalize pressure, transparent permeable skin that reduces energy cost and pressure differentials, high TMAO concentrations that stabilize water structure and proteins, genetic mutations like RTF1 that boost transcription efficiency, curved membrane lipids that maintain fluidity under compression, and expanded gene copies like FTHL27 that protect against oxidative stress. None of these adaptations alone is sufficient. Altogether, they allow life in the most extreme environment on Earth. The concentration is low, maybe 10 to 15% of brain membrane lipids, but they are there. Why? Plasmalogens protect against oxidative stress. The brain uses 20% of your body's oxygen. High oxygen use produces reactive oxygen species. Plasmalogens neutralize them. They also affect neural signaling. The curvature of the lipid influences how proteins sit in the membrane. Ion channels, neurotransmitter receptors, all depend on membrane fluidity. Plasmalogens regulate that fluidity. They did not create something new. They amplified something old. This is how evolution works. It is not invention. It is modification. The tools were already there. Pressure just demanded more of them. In 2017, a new type of snailfish, the ethereal snailfish, was filmed at 8,200 meters. It has not been captured. It has not been studied. We do not know its genome. We do not know its TMAO levels. We do not know if it has the same adaptations as Pseudoliparis suere, or if it uses different mechanisms. It exists on film, a translucent ghost swimming in the abyss, and that is all we know. In 2018, researchers found amphipods in the Mariana Trench at nearly 11,000 meters. These are crustaceans, not fish. They have exoskeletons, not internal bones. They do not use TMAO the same way fish do. But they must have their own adaptations. What are they? We do not know. The amphipods were collected, analyzed, and found to have high aluminum content in their tissues. Aluminum is toxic to most life. But these amphipods accumulated. Why? Is it protective? Is it a byproduct of their diet? No one knows. But the Peru-Chile Trench, which reaches over 8,000 meters, has no confirmed snailfish sightings. Why? Is the food supply different? Is the chemistry of the water different? Are there predators we do not know about? They reported seeing a flatfish. Later, analysis questioned this. No flatfish has been found below 2,000 meters. What they saw was probably a sea cucumber. But the question remains, what lives at the absolute bottom? In 2019, Victor Vescovo descended to Challenger Deep in the DSV limiting factor. He found plastic bags, he found candy wrappers. Human pollution has reached the deepest place on Earth before we have fully explored what lives there. The 2024 liver proteomics study. Collectively, they build a picture, but the picture is not complete. Every gene, every molecule, every structure in their bodies is tuned to function under conditions that would obliterate most life. They are not struggling to survive. They are thriving. Your bones form through genes like BGLAP. The difference is degree, not kind. The Hadal snailfish has more TMAO, more plasmalogens, less bone calcification. It is the same biology, just more extreme. How much can gene expression change in response to environment? The answers to these questions inform medicine, biotechnology, and our understanding of life itself. If we know how TMAO stabilizes proteins under pressure, we might use similar molecules to stabilize therapeutic proteins in drug delivery. If we know how plasmalogens protect neurons, we might develop treatments for neurodegenerative diseases. If we know how cartilage resists pressure, we might engineer better implants for joint repair. A 2025 study speculated that fish colonized the deep sea during the late Jurassic and early Cretaceous, 150 to 65 million years ago. This was when angiosperm plants evolved on land. The plants increased nutrient runoff into the ocean. The nutrients fed phytoplankton. The phytoplankton died and sank. The dead plankton fed deep-sea invertebrates. The invertebrates attracted fish. Fish followed the food into the abyss. Over millions of years, some fish adapted. They lost swim bladders. They developed TMAzo. They shifted to curved lipids. They became deep-sea specialists. Fish adapted. By the time the Hadal snailfish evolved 18 million years ago, the mechanisms for deep-sea survival already existed. The Hadal snailfish just took them further. It pushed the limits. Not because membranes collapse, but because genetic mutations cannot go further. The limit is not physical, it is biochemical, and that is the strangest part. Life in the deep sea is not limited by the environment. It is limited by the chemistry it uses to survive the environment. TMAO protects, but too much TMAO kills. Plasmalogens stabilize, but too many plasmalogens make membranes that cannot exist at surface pressure. Evolution has optimized these creatures for the deep, and in doing so, it has locked them there. They decrease production of calcium-binding proteins. They fine-tune metabolism in response to pressure changes. This was published in September 2024. It is the newest piece of the puzzle. Not just genes, not just proteins, but regulatory networks that adjust everything in real time. The fish does not just have adaptations. It has active systems monitoring and responding to pressure every second. The body is not static. It is dynamic. Even at 7,000 meters, even in near freezing water, even in total darkness, the cells of the Hadal snailfish are working, sensing, adjusting, surviving. A fish that lives where you cannot because its chemistry bends the rules without breaking them. That is the secret. That is the reason. And it is stranger than any fiction because it is real, it is measurable, and it is happening right now, 7,000 meters below the surface, in darkness and silence and a pressure that would turn you to liquid. There's a pink fish swimming, hunting, living, and it does so with molecules so perfectly tuned to extremity that scientists are still, in 2025, discovering how they work. The one that reframes everything. In March 2025, Chinese researchers published evidence from genomic analysis. The RTF1 mutation, the one that boosts transcription under pressure, is not unique to Hadal snailfish. It is present in all deep-sea fish below 3,000 meters. Every species, every genus, different trenches, different evolutionary lineages, same mutation, convergent evolution at the genetic level. The deep sea demanded this change. Evolution delivered. But it delivered it multiple times, independently. The mutation is not inherited from a common ancestor. It evolved separately. The same pressure, the same solution. This is not coincidence, this is biochemical inevitability. There is one way to survive high pressure with vertebrate physiology. Boost transcription. Every species that tried something else died out. Every species that got the RTF1 mutation survived. The fossil record is silent, but the genomes speak. The deep sea is not a place that tolerates variation. It demands precision. And the Hadal snailfish, with its incomplete skull and its curved lipids and its 300 millimoles of TMAO per kilogram, is precision itself.